Hello, everyone, and welcome to today's webinar, The Development of GMP-manufactured Cas9 Protein for Therapeutic Applications. I am Robert Horton of Thermo Fisher Scientific, and I'll be your moderator for today's event. Today's educational webinar seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about CTS Cas9 or related CRISPR products, please visit www.thermofisher.com/Cas9. We encourage you to participate today by submitting any questions you may have during the presentations. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. With that out of the way, I'd like to welcome our speaker, Pandhita Pochanard. Pandhita is a product manager for the Genome Modulation and Editing Group at Thermo Fisher Scientific, where she is responsible for the development of new genome editing reagents, including CTS Cas9, which is the focus of today's webinar. Pandhita, you may now begin your presentation.
Hello, everyone. My name is Pandhita Pochanard, and I'm the product manager for genome editing at Thermo Fisher Scientific. For today's webinar, we'll be discussing the development of GMP-manufactured Cas9 protein for therapeutics applications. Here are the agendas for today's training. We'll go over some introductions before talking about key considerations for therapeutic researchers. We'll then talk about manufacturing facilities and process controls, safety testing and specifications, and finally end with some performance data. Genome editing has become exceedingly popular for several applications. Due to this CRISPR-Cas9 revolution, CRISPR-Cas9 has been a significant paradigm shift in functional genomics research. It has made genome editing accessible to a wide array of researchers like it wasn't before, as the tool continues to be adopted into various workflows. Over the recent years, we're starting to see another paradigm shift with an increase in adoption of CRISPR-Cas9 technology for therapeutics development. Given the recent clinical successes and commercial approvals in this space, we have also observed an exponential growth in clinical trial pipelines as well. Shown on the graph on the left-hand side, you'll see that over the past few years, we've seen an exponential increase in trials in said pipeline. We have also seen some cell and gene therapies approved by the FDA shown on the right-hand side. This further demonstrates the promise and growth in the application of CRISPR-Cas9 technology for therapeutic development. When thinking about key steps within the translational pipeline, there are 4 distinct stages as researchers move their project from bench to the clinic, from early research and development to preclinical studies to clinical trials and finally commercialization. As researchers move their project through this pipeline, the requirements for the critical reagents needed for that project change. Researchers start to look for higher quality grade reagents to meet increased quality and regulatory requirements, and often increase volume of reagents as the demand for that project change from small scale to a large manufacturing scale. Now let's talk in more details about the 3 key requirements that therapeutic researchers look for when selecting their critical reagents to support their cell and gene therapy programs. Firstly, quality. Researchers need to know that the reagents they select can stand up to increased quality and regulatory requirements. Secondly, supply. Researchers are looking for reassurance that they will be able to purchase the right amount of reagents they need at the right time to support their programs. So it is incredibly important for these researchers that the supplier of their critical reagents will be able to produce large lot size consistently. Lastly, and just as important, performance. Researchers need and want the highest performing reagents possible. This means that not only must the reagents be high performing, but it must also have minimal lot-to-lot variation so that they can trust that lot number 1 will perform just as well as lot number 100. These are the 3 important factors that researchers look for in their reagents for their therapeutics programs, and factors to keep in mind when developing GMP-manufactured Cas9 protein. With that, we have recently launched the GIPCO CTS Truecut Cas9 protein, a next-generation Cas9 protein designed and manufactured to support clinical research programs. Our protein maintains high editing efficiency in primary immune cells and is manufactured in compliance with GMP principles. It is also manufactured to meet the requirements for the standards for ancillary materials for cell, gene, and tissue-based products. To ensure adherence to the quality and regulatory standards, we leverage an aseptic manufacturing process, and our protein is also subjected to extensive safety testing, including sterility, endotoxin, and residual host nucleic acids and protein. As the latest addition to Thermo Fisher Scientific's cell therapy systems family of products, CTS Truecut Cas9 protein is designed for cell therapy manufacturing. Our CTS product is manufactured to conform with 21 CFR Part 820 where applicable, and all at ISO 13485 certified site. CTS products are also subjected to extensive safety testing and have traceability documentation, including FDA's Drug Master files. Finally, our CTS product brands have proven use in several approved cell therapies and over 100 clinical trials. An aspect that is also critical for the development of GMP-manufactured Cas9 is in having the appropriate manufacturing facilities. It is important that the protein is made in facilities that can meet the appropriate quality and regulatory requirements. As for our CTS Truecut Cas9 product, it is made at our Biosciences Center of Excellence with dedicated GMP suites and cleanroom facilities in accordance to several regulatory directives and guidelines. In looking at the manufacturing process design and controls needed for the development of GMP-manufactured Cas9, shown here is an example schematic of the manufacturing process. To further control the risk of contaminants, a closed system manufacturing process that leverages single-use technology should be used. Additional controls from a facility management perspective is also critical. For example, all materials that enter GMP facilities should be animal origin free to the secondary level and beta-lactam free to further reduce any risk of contaminants. A few other considerations for the manufacturing process of GMP-Cas9 include a process that is capable of producing large lot size is important. As alluded to earlier, having access to a large single lot can allow researchers to use the same material lot to support that program. This could further minimize any potential variability that may be introduced when using materials from different lots. Our manufacturing process has a high yield and can make upwards of 15 grams of Cas9 protein per purification run. To further control contaminant risk, several sterile filtration steps are designed and added to the process. Additionally, in-process controls are also planned and designed throughout the manufacturing workflow as well. Furthermore, sterile filling should be completed in the appropriate cleanroom suites and safety precautions taken to further reduce the risk of contaminants. For our manufacturing process, all containers are sterilized with additional quality control in place, including performing a bioburden test on the gloves used after the assembling step. Finally, the final filled products are subjected to extensive release testing to ensure adherence to the required standards. For our product specifically, the final filled protein is subjected to 17 release tests. Again, the idea here is the controls and design that we put in place from a manufacturing perspective is done intentionally to minimize the risk of any contaminants into the actual product itself. So we take careful consideration when designing our manufacturing process to make sure that we have all the right controls and all the right steps in place to help minimize those risks so that we can meet the required standards. In addition to the manufacturing process design and controls we discussed earlier, there's also another strategy to physically segregate manufacturing steps to further minimize contaminant risk. So typically, when developing GMP-grade product, you should be leveraging both the design of the process itself as well as putting that physical layout and segregation in place as part of your design. Here's an example of the spatial layout of the GMP manufacturing suites. You'll find that a controlled and dedicated space is assigned for each key workflow step, where each activity is done in different clean room grades. You'll see that as we move closer from the left to the right to the finished good product, the clean room grade becomes more stringent, moving from a Class D all the way to a Class A clean room. And again, the idea here is to further minimize the risk of contamination. Overall, the development of GMP-manufactured Cas9 leverages both the manufacturing process design as well as spatial controls to ensure adherence to the required quality and regulatory standards. Now that we've looked at the manufacturing process consideration, the GMP-Cas9 is then subjected to extensive safety testing and release specifications. These safety testings are performed on the final finished product to ensure that it passes all the required specifications. Here at Thermo Fisher Scientific, our CTS Truecut Cas9 protein is subjected to 17 QC tests performed on each finished good lot before release. The release specifications can be categorized into 3 distinct groups, each measuring for different source of potential impurities or contaminants. The first group of assays are product-related impurities. These include identity and purity assays that measure for any product-related impurities. Second group of release testing include process-related impurities as well as safety. These assays measure the level of contaminants from various sources, including enzymatic, host, and process. Finally, you have characterization. These assays confirm the characteristics of the final product, including concentration, pH, and activity. Together, these release tests help to ensure that the final GMP-manufactured Cas9 product meet all the specifications and requirements in accordance to the appropriate standards for cell therapy applications. Now we're going to move into our performance data. Here we evaluated the performance of our CTS Cas9 protein against our research-grade Cas9 product. The goal is to assess the effect of GMP manufacturing process and controls on product performance. We selected primary T cells as our model of choice to demonstrate the application of CTS Truecut Cas9 for autologous T cell or CAR T therapy workflows. From an approach perspective, first we evaluated the protein activity with an in vitro cleavage assay. Next, we evaluated the performance of these proteins in primary T cells at the small neon electroporation system of 10 microliter. Then, we scaled up our experiments by 10-fold using the larger scale neon electroporation system. First, we look at the protein activity using an in vitro cleavage assay. The benefit of this assay is where we are strictly only looking at the protein activity via cleavage activity level. We then diluted the RUO and CTS Cas9 proteins to a linear range and measure cleavage activity accordingly. You'll see on the left-hand side in the graph that across our tested protein dosage on the x-axis, the activity between the RUO and CTS Cas9 proteins are comparable. This result suggests that CTS-Cas9 was able to maintain in vitro cleavage activity across doses relative to the RUO Cas9. Next, we wanted to confirm the activity of the CTS-Cas9 protein observed in vitro and to make sure that it is still preserved when transferred to a cellular environment. As mentioned, we selected primary T cells for these experiments as they offer the appropriate cellular context needed for CAR T cell therapy development workflow. We selected 7 guide RNAs targeting 4 CAR T cell related therapeutic genes. These guides include those used in literature as well as some other in-house selected options. For this experiment, we leveraged the standard or small benchtop scale neon electroporation system of 10 microliter. We looked at cell viability and editing efficiency from the 2 proteins. When looking at the cell viability shown on the left-hand side here, we observed that both proteins yielded high viability or low toxicity. Then we measured the editing efficiency using next-generation sequencing and saw that both RUO and CTS-Cas9 proteins perform comparably from a performance perspective. These results suggest that our CTS-Cas9 proteins have comparable functional performance relative to the research-grade Cas9, and that the GMP manufacturing process did not negatively affect performance. Wanting to evaluate the CTS-Cas9 performance further at a larger scale, we also evaluated both proteins at 10x scale-up using the larger neon electroporation platform of 100 microliter. Once again, we look at cell viability and editing efficiency. This time, we selected 4 guide RNAs from the previous 7 to do further analysis. Similarly, you'll find that the cell viability shown on the left-hand side once again is comparable between the 2 proteins. Then for editing efficiency, in this specific experiment, we wanted to evaluate both indel and HDR from both proteins. HDR, or homology-directed repair, is the pathway that is important for therapeutic researchers as it enables more precise edits. For HDR assessment, we use single-strand oligos as our donor, and for our electroporation, we use a genome editing buffer that is optimized for knock-in application. Across the 4 guide RNAs we assessed, we were able to achieve high HDR percentage for both proteins. Then when looking at the overall editing efficiency, shown as a number on top of the graph right here, you'll see that both proteins once again achieve comparable efficiency across all targets at the 10x scale-up. These results further confirm that GMP manufacturing process did not have a negative impact on performance. It further confirms that high HDR percentage can be achieved with the CTS-Cas9 protein at large scale. Finally, we assessed the quality and performance of the CTS Truecut Cas9 across 3 different manufacturing lots. Here we performed the experiment in primary T cells targeting 2 genes and using 2 guide RNAs. Serial dilution was performed to better interrogate the editing efficiency achieved with decreasing protein amount. At the first target, track 4, shown on the left-hand side, you'll find that comparable editing efficiency is observed across all 3 manufacturing lots throughout the dilution range. We also saw a similar trend and performance at the second target as well, shown on the right-hand side, or CD52-6. Both these results showed that consistent lot-to-lot performance was achieved. This is a direct indication of a very tightly controlled manufacturing process, which we talked about earlier. In summary, there are 3 key requirements that therapeutic researchers look for when selecting their critical reagents. Quality. Once again, we talked that robust manufacturing process and controls is necessary to make sure that we keep safety risk to the minimal. Secondly is supply. Today we talked about the importance of high yield manufacturing process. which would help minimize risk of performance and variability and also assist in supply continuity. Lastly, we talked about performance. As mentioned, high-performing reagents with minimal lot-to-lot variation is important for therapeutic programs. These requirements are the key drivers when designing the manufacturing process or the development for GMP-manufactured Cas9 for therapeutic applications. With that, I want to thank everyone for your time, and we will now open the floor for questions and answers.
Great. Thank you, Pandhita, for the very informative presentation. We will now start our live Q&A session portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen and go ahead and hit send. And we'll answer as many questions as we have time for. With that, let's go ahead and get started as the questions are starting to come in. So Pandhita, I know that you also have a research grade version of this Cas9 protein. Can you expand a little bit on the differences between that research grade and the CTS grade product?
Yeah, that's a really good question. So I know I kind of touched on this as well in the presentation, right? So just to confirm that both of our RUO and CTS Cas9 proteins are the same protein from a functional perspective. We saw, I just showed you just in a couple of slides ago that both have comparable performance. Right, but so the differences, the key differences here is that with the CTS Truecut Cas9 protein, with that additional sort of values coming from the CTS brand, we offer more documentation to meet regulatory requirements. And as you saw as well, right, the protein itself is also subjected to tighter quality controls, including more process controls, extensive safety testings, as well as release specifications. So that's sort of the key differences, I would say, between the RUO and the CTS Cas9 protein.
Good, thank you. I know you tested some T cells with the CTS Cas9. Did you test any other cell types besides T cells?
Yeah, and I feel like I should have mentioned that too in the presentation, right? But as you saw, so the data that I showed today are all in T cells. But we have also tested the CTS Truecut Cas9 in iPSCs as well. And the intent of that was to mimic kind of its use in that allogeneic workflow, because we talked a lot I talked a lot in this presentation about autologous. So we did tests in iPSCs. But as mentioned, again, the primary reason why I chose to display primary T cells was because it's our model of choice for its representation of autologous T cell application or CAR T cell therapy development workflow. But we did, as I said, we also tested in iPSCs as well. Yeah.
And I noticed that you also used the Neon electroporation platform. Have you guys tested any other platforms or any more high throughput versions?
Yeah. So so we tested in obviously the 10 microliter and the 100 microliter, and the purpose of that was just to kind of mimic our use case of that small scale electroporation. But we did also kind of to answer that question, right? We also did think about large scale up. So we also have tested the CTS Truecut Cas9 in our CTS Xenon electroporation system. And for those who may not be very familiar, our Xenon electroporation system just launched last month, and it's our large scale electroporation platform. So we also did experiments on that platform as well. I didn't show that data today, but we have that information, and we've kind of tested it and confirmed that the performance didn't drop as we change our electroporation sort of volume or conditions, which is great.
Great. I think that high volume electroporation is going to be important for this application moving forward. So very good.
I think especially for therapeutics application, right? If you think about it, it's like you're going to want to do the small scale experiments, which is where the Neon comes in perfectly before kind of scaling that up to the Xenon. So having access to both platform and having reagents that work well on both platform, I think is going to be really important for therapeutic applications.
Yeah. So there's a question here about stability. So I know in your presentation, it looks like you showed some data about some of the lot-to-lot variability of the CTS Cas9. Maybe you can comment a little bit more on stability on the CTS Cas9 protein itself. Any other data or experiments you guys performed?
Yeah. So at time of launch, we have real-time stability data to support 12-month shelf life. And currently, we're testing additional time points. So those are being collected right now with the intent of, assessing for a potential shelf life extension in the future. But just at the time of launch, as I said, we have 12 months. And that information is actually going to be on your C of A as well when you purchase the product. And then as far as additional stability experiments, we did do some short-term stability experiments as well on the CTS Cas9 RNP, which, as you recall, RNP is when you complex the protein with the guide RNA, right, in various conditions. And one of the example sort of use case that we leveraged was to leave that CTS Cas9 RNP at room temperature for an extended period of time. And again, the thought here was that we wanted to mimic what researchers would do in situation where you're doing like high throughput workflow. So from this experiment, we actually found that our CTS Cas9 RNP is quite stable. And again, I think that speaks to the manufacturing process and the controls that we put in. We just were able to achieve that kind of high purity product, making it quite stable at room temperature for an extended period of time. I think our time point goes up until like 16 hours, which is obviously kind of what we wanted to mimic, as I said, in case researchers are doing high throughput workflow and they need to leave those reagents out on the bench for a longer time.
16 hours. That gives them a lot of flexibility to design your experiment and use it. That's great information.
Right. Exactly. And that was why we asked that questions and wanted to do that experiment to see, what that would look like, just mimicking a couple of different use conditions. Yeah,
excellent. There's a question around, is the drug master file available on the CTS Cas9 protein?
Yeah. So as I said, right, this is kind of part of the value of our CTS brand product as well, in that we offer that additional documentation. So for the CTS Cas9, yes, the drug master file will be available. So that's something that we also have a process in place if any researchers wants to request it. There's a kind of a almost like a form or a form fill for them to fill to request that drug master file. But yes, we do have that, and we will have that available for this product.
Great, excellent. And there was a question from a Jenny, and sorry, I didn't quite understand the question, so maybe if that person could resubmit their question, I'll be able to ask it to Pandhita. But in the meantime, while they are typing that, maybe you could just tell us a little bit more about where they can get more information about the CTS Cas9.
Yeah. So obviously, as I said, we just launched this product a couple of weeks ago. So if you go, on thermofisher.com, you're going to be able to find a lot of information on CTS Cas9, as well as some of the additional data actually will be on when you type the product into the search bar on thermofisher.com. You'll be taken to our product page, and actually on that page, we also have additional data that I didn't show today. So some of the iPSC's data will be kind of available there as well. And so, I feel like that's probably the best place to get all your information. And of course, we also have a number of application notes as well on this product that if you're interested, you can also find that online on thermofisher.com. But also, if you go on thermofisher.com and you don't see what you're looking for, please feel free to reach out to me directly as well, and I can direct you to the appropriate team or get you the information that you may be looking for.
Right. And then there's a question that came in regarding the RNP complex, and I think they're talking about, must have been when you were talking about the stability studies and everything, and they want to know a little bit more about how you prepared that complex. So maybe you could just expand a little bit more on that.
Yeah, so as far as preparing that RNP complex, actually, so I just wanted to point out as well, we followed the protocol that we have. So we have a user manual on this product online, and it's our standard protocol as far as, kind of mixing the Cas9 with our synthetic guide RNA to form that RNP complex. So if you would like additional information on like the exact protocol, the ratio that we use, I would actually recommend going to that user manual just to get the exact numbers. But that's sort of the process that we followed. I don't want to go into too much detail today, but you know, like I said, the exact information and the protocol that we use for the preparation of the CTS Cas9 RNP is in the user manual with the exact ratio, as well as our electroporation conditions that we use on the on the neon is also in that user manual as well. So certainly something that I can share with you directly, and you'll be able to sort of you know, see all the different conditions that we use for this experiment and the data that I showed.
Great. Thank you. Just waiting to see if any other additional questions come in. That looks like it's about it. So anyways, all right. With that, so wanted to thank you again, Pandhita, for your time today. You know, we'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time to answer today, or we will follow up afterwards, and we'll definitely reach out to you. The webcast can be viewed on demand. LabRoots will alert you via email when it's available to replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until then, thank you everyone for your time today, and have a great rest of your day. Thank you.